अब हम बात करेंगे इलेक्ट्रोफोरेसिस प्रोसीजर की टू सेपरेट द प्रोटीन्स ऑन द बेसिस ऑफ मॉलिक्यूलर वेट। अगर सारे प्रोसेस को हाईलाइट किया जाए या सीक्वेंस में डायग्रामेटिकली एक्सप्लेन किया जाए, देन वी हैव दिस स्कीम। सबसे पहले जेल कास्टिंग है, देन वी आर गोइंग टू लोड दीज जेल्स इनटू द इलेक्ट्रोफोरेटिक टैंक। We are going to add the buffers, anode buffer and cathode buffer. Then using Hamilton syringe, we are going to load our samples in the wells. Next would be application of the electric current to do the electrophoresis, and then at the end we are going to get our gel with proteins separated on the basis of molecular weight. स्टेप बाय स्टेप अगर बात की जाए तो द फर्स्ट स्टेप इज़ टू माउंट द जेल्स। हमने जो जेल्स कास्ट की थी वो हमारे पास जेल कास्टिंग स्टैंड पे मौजूद हैं इन बिटवीन द टू ग्लास प्लेट्स। वी आर गोइंग टू टेक दोज ग्लास प्लेट्स अलॉन्ग विद द जेल एंड वी आर गोइंग टू टेक दैम इन द इलेक्ट्रोफोरेटिक apparatus. Then we have two areas. One is the cathode area, the second one is the anode area. We are going to fill them with the electrophoretic buffer, and just to ensure that the anode area, which is a smaller area in the center, cathode area which is in the peripheral region, it should not be leaky and the buffer should stay in there, otherwise electrophoretic process will automatically stop. Once the glass plates along with the gel are in the electrophoretic chamber, now this is the time to prepare the samples. Samples can be prepared with the known concentration so that we have equal amounts of proteins which are subjected to electrophoresis, and then we can study them subsequently. We can use 10, 20, 40 microgram or whatever amount of the protein you like on the basis of previously obtained concentration can be prepared along with the sample buffer. जितनी वॉल्यूम हमारे पास मौजूद है प्रोटीन सैंपल की, वी आर गोइंग टू ऐड एन इक्वल वॉल्यूम ऑफ सैंपल बफर। यहाँ पे इस चीज़ का ख्याल रखना ज़रूरी है कि वेल्स की एक लिमिट है टू टेक अप ए स्पेसिफिक वॉल्यूम। सो वी हैव टू मेक इट श्योर कि अगर हमारे पास जो प्रोटीन सैंपल बहुत ज़्यादा डाइल्यूट हैं, we need to precipitate them and we need to concentrate them and we need to restrict to a specific volume that can be housed in the wells of the gel. At the same time, we need a pre-stained standard or marker for protein molecular weight, and sometimes we can also use a protein sample which is of our interest as a positive marker which can be run in a separate well. So the next step would be, after placing your gels in the gel electrophoretic chamber, after filling them with the electrophoretic buffer, now this is the time to take up the comb. It is advised कि comb को दोनों edges से पकड़ के gently pull किया जाए। किसी एक एज से पकड़ के या सेंटर से पकड़ के अगर पुल किया जाएगा तो एक जो वैक्यूम क्रिएट होगा, उस वैक्यूम से वेल्स की शेप खराब हो सकती है। सो जस्ट लुक इन दिस पिक्चर, वेरी जेंटली होल्डिंग फ्रॉम द टू एंड्स, द comb is going to be pulled up, but before pulling up the comb, make it sure the buffers are already in there. The chamber should be filled up with the buffer. Then sample is loaded using the Hamilton syringe, using the micropipetter. In order to ensure the presence of the wells, we can use a well guide which can be seen as a yellow comb-like structure in this picture with the researcher. अगर हम syringe use कर रहे हैं, Hamilton syringe use कर रहे हैं, तो in between the two samples, the syringe should be rinsed with the buffer so that there should not be cross contamination. One protein sample should not contaminate the other protein sample. Once the sample loading is complete, we need to clean this syringe because we have to use it again, and this cleaning can be done with water and subsequently with ethanol. Once the sample loading is done, cover the apparatus, close it and subject it under current 160 volt for electrophoresis for some time. The time can be variable just keep an eye on the blue marker in each well. That is going to mention or tell you how far our gel has run in the electrophoretic chamber.